So I've already gone over some of the main characteristics of the bacteria, the domain bacteria and the domain archaea, and we are now moving into the domain eukarya, which will really be the whole rest of the organisms that we talk about throughout the whole course. So we're going to start off with a group called the Protists now. It used to be kingdom Protista, but that is currently really not the case, but that's traditional. And so as I break down this lecture into multiple parts, I'll be this one and then a few others that follow it, looking at some of the different characteristics and some of the groups in particular within the Protista. First, I'm going to point out this sort of phylogenetic tree here, which is sort of a rough. I'm not linking all the groups and all, and I'm not putting the main names to the categories of the groups because it's something that changes a lot. So I want to say what we're going to focus on are really specific characteristics of the Protists, things that many of them share that are unique and that you're going to find as being definable and the same. The classification, again, is largely based on gene sequencing, which, again, is very difficult to say, let's study a group with this name that has this particular sequence. And here's another group of organisms that looks exactly the same and does most of the same stuff, but because of a sequence, we don't have them being very closely related to one another. How do you organize that into a lecture that makes any sense at all? It is really difficult. So we're going to be organizing things more by, again, common structures, functions, physiology, and more the traditional way that the organisms were once categorized into groups. Because these things make a little more logical sense in terms of learning about the organisms themselves. As far as their evolutionary relationships and classification, that is sort of, we're going to treat that slightly as a separate topic. It's also a topic that continues to change. There's not going to be a great point in, if you look in your book, each of these are groups of organisms and they each have names associated with them. However, those names are recently assigned, not necessarily accepted by everyone, and are highly likely to change within the next few years. And so depending on, even when someone watches this particular lecture, if I were to put names there for these groups, those names may not exist at the time that you would watch it. So what I'm going to talk about are the core groups of organisms who have names based on characteristics that are not necessarily going to change ever, and then where they fall out in here, that's something that's going to always be updated. So you'd have to kind of look at what's the most recent thought on how they're classified. So what I'm going to do is give you a brief overview of this, and I'll kind of refer to it back as we talk about other sorts of things. What we're seeing here is a eukaryote ancestor. So an ancestor of the eukaryotes giving rise, and this is everything that's a eukaryote. Right, now, within this, what do we have? Well, we have down here, oops, let me get another one up. We have the animals, right, the group that we belong to, and we also have the fungi. So we have two kingdom Animalia and kingdom fungi are both down here on the same sort of branch together. It's telling us a couple things. One, they're closely related to each other. They're probably more closely related than the fungi are, again, to plants, which we're going to find up here. And then there's another group, a group that is typically considered a Protist group, and it's right in there with them. So essentially, this group of Protists, what we're saying, is more closely related, say, to animals than they are to the other Protists. Right? So that's why Protista isn't like it's considered its own group right now. It's really what's called a paraphyletic group. So you see, if we, so I'll write that down here, since we've talked about cladograms in the class, monophyletic, it's not paraphyletic. The paraphyletic, or the reptilia, this is really a polyphyletic group in many ways, because also there's probably many different ancestors for some of these groups that are not the most recent common ancestor, and we're also removing members that could be in groups and putting them into other groups. So There's aspects of it that it's paraphyletic. There's a whole larger scope of it that's probably more polyphyletic as well. Definitely not a monophyletic group. That's the main thing. So here we have animals and fungi down here. Here we have land plants, so kingdom Plantae. So generally, the eukaryotes were broken into 4 subgroups. Okay, those would be kingdoms: Animalia, kingdom Plantae, kingdom Fungi, and then kingdom Protista. All right, so we still have animals, Animalia, and for the most part, they're going to be monophyletic groups, a group themselves. And then we talked about the reptiles a little bit when I talked about cladograms, and there's a couple things within them that are a little bit need modification, but overall very good alignment with the relationships that we know and structures and these sorts of things. Same thing with plants for the most part, and same thing with fungi, although fungi there's a little more movement in there. When we get to the Protists, really all over the place, and they have been for a very long time. Part of the problem is just so many different groups. And what you're going to find is as we start to talk about some of the actual structures and characteristics, you're going to see that they're really going to be spread out throughout all these different groups as well. So what do we have? We have a group of Protists up here that contains Euglena, Euglenozoa, and Parabasalids. There's another group that contains that have flagella. Now we have dinoflagellates, ciliates, little diatoms, which are photosynthetic, single-celled, and then brown and golden algae, which could be multicellular. So we have single-celled, multicellular organisms. We have some with cilia and with flagella, and they're all in the same group. Here's a group called, I'm not giving you the name of the group, but it has other members as well, but 2 of the main ones are Foraminifera and Radiolaria. So these are amoeboid-like organisms, so single-celled organisms, amoeboid in terms of their not a uniform shape and the way in which they move, but they also form shells. Some of them can secrete shells, many of them secrete a glue, and they glue together particles to build a little shell for themselves. It's something that makes them unique. So that group in a way is, in some ways, is actually the most specific, including where the members share certain characteristics that members in these other groups sort of have, and it makes them a little more unique. Here we have photosynthetic organisms, red algae, green algae, and the land plants, now all in this particular group. We have slime molds, which I talked a little bit about with multicellularity in a previous lecture, and amoeba, so again, single-celled amoeboid, but the slime molds having a multicellular aspect to them. And then finally, in this bottom group, I talked about animals, fungi, and these choanoflagellates. So really a whole bunch of different things like actually all put together into that group. And there's another classification scheme that has 5 instead of 6 and actually puts all these together in a different group. So as I said, the names of those larger groups is not something you're going to really need to know. And like I said, I'll refer to this at times, but what we're really going to focus on now is characteristics, and we're going to start off with some of the main characteristics of Protists, and then some specific structures. And some of these structures are going to help us actually carry over to studying other groups as well, and this is where we're just going to start with it. So, Protists, mostly single-celled organisms, but there's some multicellular. So a lot of these are single-celled organisms. As far as motility goes, we see a whole variety of different types of movement in them. Now, in terms of structure, we'll kind of take a step back. Cell membrane, very much animal-like, but some of them have a cell wall, and that is much more plant-like if they have one. So they do have the typical cell membrane, and then they can have a cell wall. The cell membrane is similar to that in all other eukaryotes. The cell wall is a much more plant-like. Not like the archaea, not like the bacteria. It's something that's the Protists and the plants kind of would share in common. Not all of them are going to have the cell wall. Now, in terms of movement, so that we have a cell, and then we have the movement of the cell, and I talked about for the others with a flagella. What I'm going to bring in here is a discussion of something called a cytoskeleton. And the reason for that is that it's something that is highly conserved. So this is called what referred to as being evolutionarily conserved. So we can find a structure in one organism, and it's very specific in terms of the proteins that make it up, the arrangement of those proteins, how they all come together, and we find that same structure in a different group of organisms and another group of organisms and another group of organisms. So for example, animals, we have some cells that have flagella, right, and some cells that have cilia. Flagellates have a flagella. Fungi can have cells that have flagella to move around. These other organisms have, some of them have cytoskeletons. Some of them, again, they get kind of blurred in terms of, like, some of these have cilia and flagella, and some of them don't have any cilia or flagella in terms of motility. But if they have one of these structures, it's going to be the same as it is in all the other groups. So it's something that's very uniform across all these different groups. And so that's what we're going to talk about it. Now, you will read that the eukaryotes have a cytoskeleton and the prokaryotes, so the archaea and the bacteria, do not. They do not have a cytoskeleton. And that is not completely true. It's all the way in which you interpret that terminology. The archaea and the bacteria have internal proteins in the cytoplasm that can form this ring called a Z-ring, that can then help in binary fission and cell division, and the movement with a structure called a divisome that moves and separates the bacterial chromosomes into two. So like the movement in mitosis of the chromosomes, sort of like that, but with totally different proteins doing it in a totally different way. So do they have internal proteins that form structures that create movement within the cells? Yes, but they're totally different proteins than what we have, and they do it in a different sort of way. Okay. What we're going to talk about here is going to be the same for pretty much all the eukaryotes. So whether we're talking about the euglena or an amoeba or a fungi or a plant or an animal, all going to be having these same structures in the exact same way. Right? And refer to this cytoskeletal structure as being something that has microtubules, microfilaments, and then something called intermediate filaments. The microtubules are made up of protein called tubulin, and that's what we're going to be focused on right here. Okay, the microfilaments are composed of a protein called actin, and we're not going to talk about this right now. And the intermediate filaments are made up of other sorts of proteins. So we're not going to focus on those at the moment. We're going to be focused on the microtubules. Okay, so this is part of the cytoskeleton, common for all the eukaryotes, including the Protista, and we're going to talk about it with the Protista specifically because many of them are single-celled organisms that move around with a cilia or flagella, and traditionally, the way in which this group was classified, so when it was Kingdom Protista, we had flagellates, ciliates, and amoeba. And so at one point in time, they were classified by their method of movement, right? Whether they had a cilia, a flagella, or they moved by amoeboid motion with pseudopodia. Right? And you can see that's not really the case now because you have ciliates and flagellates in the same group, and then you have flagellates in another group, and you have flagellates in another group, so they're kind of spread out, all over the place. That's not the way in which they're done now. But we're going to focus on what are these structures and what's the commonality, because that's not going to change over time no matter who or what type of sequencing is done. So the cytoskeleton is made up of all these different things, but the one part of it we're focusing on are microtubules. Now, microtubules are made up of a protein called tubulin. And there's alpha tubulin and beta tubulin. So two different types of tubulin. primarily. And again, there's some variations to this, but generally this is always going to be true and the same. Those two tubulins come together and form a structure called a dimer. Like this, where an alpha and a beta are together. And then the alpha-beta dimers join with others, and they form a filament. And so this filament is essentially repeating alpha tubulin, beta tubulin, alpha tubulin, beta tubulin, alpha tubulin, beta tubulin. So it's just tubulin, 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 tubulin protein with some variations to it. So it's just long strings of this particular protein. Then what can happen? These filaments can then be arranged into tubes. And these tubes are then referred to as the microtubule. So you can see like this would be, say, one filament here, and then you have another filament, another filament, another filament, another filament, and they kind of then make this tube, hollow tube-like structure. And that is the microtubule. Now, cilia and flagella are both made up of microtubules. And in fact, cilia and flagella are the exact same structure, except the only difference is their length. So for an organism, this is a single-celled organism, and it has cilia sticking off it. They're short. If it has a flagella sticking off it, it is long. But if you were to slice this flagella, take a look at it, like what does it actually look like in a cross section, and you were to slice a cilia and take a look at it and sort of compare them side by side, for both of them, you're going to see this. And this is a structure called a 9+2 arrangement of a cilia or flagella cross section. So this is sort of the cross section all right, so the cross-sectional arrangement of both cilia and flagella. And so what are you looking at? 9 what plus 2 what? So this is a tubule, and so that's a tubule, okay? So these are doublets. So they're two tubules together. All right, so we take a tube like this, then take another tube, so like if these two things here, and put them side by side and connect them to one another. That's what these things are here. Right. And now we have, if you were to go around this circle here, 1, 2, 3, 4, 5, 6, 7, 8, 9, there are 9 of them, forming a ring-like structure. And then this is hollow. Again, so these are hollow. So you have to keep in mind, we're expanding out the structure. This is a single microtubule, okay? And then the microtubules themselves sort of form like another tube, essentially, as they assemble. So it's like a tube within a tube. And in the center of this are 2 more microtubules, not set up the same way. They're sort of independent. There are proteins that link these together, like this. And then there are proteins that link these doublets to the core, like little spindles. And then there are proteins that link these together, like this. And this then forms the wall, the actual structure of the cilia and or the flagella. Now, how do they actually move? Well, when we looked at the so this is, say, the cell membrane. And we'll have this be the, we'll just have it be a celiac, so we'll zoomed in on it. there are going to be proteins along this that are going to be able to change shape. These are proteins referred to as dynein. We're not going to go into all the biochemical details here, right? So you might see that or read about it. So the dynein proteins are going to change shape. They're going to cause a sliding motion, and what it's going to do is somewhat then say pull down on one side and push up or extend on the other side, and then it's going to cause that structure to kind of tilt in one way. And then these structures can then, the proteins will alter their shape and then move back the other way, and so the structure will tip back then, and it will be able to kind of move back and forth like this and flap. And so the short structures, like a windshield wiper, will just move back and forth as it kind of gets shortened on one side and then lengthened on the other, and then that side will shorten and the other side will lengthen, and it'll just move. If this were a very long structure, the flagella, then as these motors are moving at the base back and forth, the longer parts toward the tip are going to start to whip back and forth, and that creates more of this whipping motion of the flagella. But structurally, they are going to be the same. So this is the same exact structure we find for movement in the parabasalids and the euglena, in the dinoflagellates, and in the ciliates. Any modal algae that we have often modal stages of them, we're going to find them there as well. All these algae here that are modal, we're going to find them there. We're going to find them in land plants who have sperm that swim around in water, and they're going to have the same exact flagella. We'll find them in the fungi and the coniflagellates and the animals, all the same structures. Okay, the same layout, the 9+2 arrangement of microtubules, and the tubules made up of the same particular proteins and the movement in the same particular way. It's all essentially going to be the same. All right, so going back to the classical ciliates and flagellates, we have those as two different groups. However, you could see, at the core of it, there's really the same structure, and that's why potentially having ciliates and flagellates in the same group today isn't as odd as it may seem, because we can see they're talking about the length of the structure generally as well. Organisms that have flagella typically have few of them. Organisms that have cilia usually have many of them. So a ciliate, an organism that has cilia covering the entire cell, that's just the cilia typically covers the entire cell. So the cilia go all the way around, the entire cell, right, like this. Organisms that have flagella, or single-celled organisms that have flagella, may have a single flagella like this. They may have a tuft of flagella. They could have flagella at both ends like this, or they could have flagella, all the way around. And bacteria have similar kinds of arrangements, but they're totally different kinds of flagella. They're made up of completely different proteins. The motor that makes them move is completely different. Their motion is completely different. They spiral, they spin around, whereas these do not. They move back and forth. So the flagella of the bacteria is not at all related to these Protista and eukaryotic, flagella. But the flagella in all these groups is essentially the exact same structure. So kind of keep that in mind. And that's kind of just the first part, talking about motility within the ciliates and the flagellates, not as groups, but just as sort of types of single-celled protists that move around with those. Or for the multicellular organisms like us that do have structures in our bodies that are cells that are covered in cilia, we find we also have the same structure for our cilia as well. To finish up here with just movement and motility, I'm going to add in one other type, and that is what was one of the classic groups used to be historically of the Protists were the amoeba, who were categorized essentially by not having a particular shape. Okay, so the cells, an amoeboid shape essentially means shapeless, or something that can constantly change or alter its shape. So if this is a cell, this is the cell nucleus, let's say here, this extension of the cell is referred to as a pseudopodia. And if later on you're wondering, like, what kind of questions I'm going to be asked about any of this type of thing, things like, how do single-celled eukaryotic organisms put into the Protista move? You know, cilia, flagella, and pseudopodia, which are 3 structures. Which of those, cilia, flagella, and pseudopodia, use microtubules? It's the cilia and flagella. Pseudopodia now are going to use other types of cytoskeletal structures. They'll use a little bit more of the actin. Now, they do have some microtubules involved in it, but it's a lot more with these actin proteins. So what's going to happen here is they're going to be organizing centers that are going to build structures within the cytoplasm of the cell, and those structures are then going to push onto the cell wall, and they're going to start to make the cell wall push outward like that. And so that will extend the cell wall. Sorry, I keep saying cell wall. Cell membrane. Some of these organisms can have a cell wall, but usually it wouldn't be so easily manipulated like this, not as flexible. So it's really just the cell membrane that would be a much more flexible structure. And again, we're talking about the membrane of a eukaryote, which is the typical phospholipid bilayer structure. So the actin filaments and other types of filaments inside the cell can push on the membrane, distorting the membrane, and then creating extensions of that. And then those extensions can then be used to create adhesive spots, so essentially where they might actually like stick to one particular location, and then they'll shorten. So then these structures will move in the opposite direction, shortening themselves, and they could pull the whole organism, you know, to a new location where the organism itself now moves to that particular spot. So they can sort of crawl across the space by extending out a pseudopodia, sticking to it, and pulling themselves and moving across an area. So amoeboid organisms, like there's several different groups of amoeba, the slime molds, the single-celled organisms within that group move through amoeboid motion. Forams and radiolaria, they are amoeboid cells that move around through amoeboid motion. And many types of animals have amoeboid cells within their bodies that move around the same way. So that's just something slightly different, still using a aspect of proteins organized in a specific way in the cytoskeleton, but different sorts of proteins in their internal, within the cytoplasm, pushing and distorting the cell membrane. And so again, we're not going to go into all the chemical details of all the adhesion and everything else. It's just the basic overview of this. So for right now, so like I said, we're going to break down Protists into multiple short lectures. And so the first one is just kind of introducing the group and saying it really doesn't have a taxonomic level at this point. So like eukarya is a domain. Animalia, fungi, plants, those are kingdoms. Okay. We used to have, kingdom Protista, which would then be, all these others, which again would make it a para and in this case maybe polyphyletic organization of the group. And so recently, because of other genetic evidence, people have said we should just sort of abandon that whole thing. But then what do we call them? Some people refer to the term supergroup, or are there going to be phyla breaking out here? This is something that's going to be changing most likely as the future moves along. So instead of calling it by any particular name right now, that's probably going to change anyway. We're just going to refer to the members of this group are essentially not plants, not animals, not fungi, okay? But they are eukaryotic organisms. They are mostly single-celled organisms, but we can also have multicellular organisms. As we go through some aspects of physiology, we're going to find some are photosynthetic, some are not photosynthetic. Some do have different pigments for photosynthesis. Some move and some don't move. Some move with different mechanisms. Some have cilia, some have flagella, Some don't have those. Some move through amoeboid motion. Some don't move, at all. They can move through having little gas chambers inside them, inside the cytoplasm, that they can use for buoyancy. So some of the ones that live in the oceans can actually regulate where they are in the water column. Based on that, on things like that, like a scuba diver would regulate their buoyancy, the cells can actually regulate that. So they can have some control over movement, but it's a very different sort of thing. A lot of diversity really within the group, and that's why it's so broken up. So we'll continue on next, talking about some of the different types of nutrition and metabolism, physiology aspect, of the group. What do they do for photosynthesis and nutrition, the heterotrophic ones, and how they phagocytize other cells. That's the sort of the next